In this video, I'm going to walk you through some of the things that you can do for week two's big assignment. So every week in this class, an intro to programming for bioinformatics, I'm trying to give you guys an opportunity to create a public-facing website that you could put into your portfolio. So this week, I wanted to give you a data set that was really big and kind of complicated, and lots of different options for how you can tell a story with that data. So I've left it pretty wide open. If you get stuck as you're working on this and you're like, I have no idea what kinds of things I want to do, I'm going to show you some of the plots I made in this video. And you're welcome to use some of those plots and just vary the colors. If you have an idea of something you do want to ask, like a research question or a hypothesis, you can also choose to do a different plot. Any three plots from this data set that are well formatted and tell a story will earn full credit. So the data set that we're going to start with is gene quantifications that are in a tab separated variable file. You can see this file that's just text is 10.5 megabytes. So it's a really large file, and we're going to want to download it on our computer and not open it so that we make sure that we don't make any formatting changes before we bring it into R. Once we have the file, we want to have, best practices. We want to make sure that we've started a new project. And so what I've done here is I've just on my desktop for this course, I have a new bin 501, week 1, week 2 project for every week. That way, I don't have to have a million different project folders, but I stay organized and I have some idea of which packages and which libraries I've set up to be active in each of the individual projects. So you see from the earlier homeworks, I have the Diamonds data set and the World Happiness Report data set that we did for tutorials. And I have ENCFF 166sfx.tsv, which is that file from ENCODE. So I have nice file for gene expression, and it's a really simple file format. One of the things I can do in our studio just before I open it up is I could click on it, but one of the things that I want to warn you about is that it will give you a little bit of a hard time with opening it because it's too big. So let's just try reading it in. It's tab separated, so read.table is going to be our best option for reading it in with almost all the default settings, but it does have a header, so I want to make sure I set header equal to true. And when I read it in, I called it my GEX for my gene expression. I can see all of the types of data that are in the file and all the columns of data that I could use as inputs for a plot. One thing that's really nice about this ENCODE data is it's already tidy. We only have one observation, one gene ID per row. And so because of that, we don't have to do anything to be able to use this in ggplot. So we have each of these gene IDs, the transcript IDs, the length of the gene, the transcripts per million. So transcripts per million reads in the sequencing run, fragments per kilobase per million reads. So this is a little bit of a better measure of gene expression. One of the things that we're doing is we're taking each of the data points that we get, so however many observations there were for this gene in the sequencing data, and we're making sure that we are going to have a really good amount of data that is proportional to the length of the gene. So we're dividing by both how many million reads there were, right? Like how big the run is should affect how many reads map to each gene. But we're also dividing by the size of the gene. If you don't do this, if you just look at TPM, what ends up happening is the longest genes in the genome end up having the most fragments aligned to them. So genes like Titin end up getting lots and lots of hits just because there are a lot of nucleotides long. FPKM is nice because it accounts for that. And by dividing by the size of the gene in kilobases, you have a better idea of the relative expression of the gene and not just like how long is the gene. So you have all this different information that we can look at. We can look at FPKM statistics. So you could do some really nice plots using those pre-calculated statistics. But the main information is going to be the gene ID and the FPKM. Once you've read it in, you can start to try out different plots. So the first thing I did was I wanted to look at gene length. I was really interested to see like, well, how many genes are very large, how many genes are sort of like in a typical range in this data set of human genes. And we can see that there are very few genes that are 200,000 base pairs long. Most of them are really like in the 0 to 5,000 range. The next thing I can do is also look at length, but bin it a little bit differently and only pay attention to those smaller genes. So what am I doing? I'm looking at the things that are less than 10,000 base pairs long, because when I looked at all of the lengths, most of the genes were in that range. So before I made my plot, I filtered. And when I do that, I get a histogram where I get a better picture of what's happening with gene length in that smaller portion of the graph. I can also plot FPKM for the top genes versus their ensemble gene ID. One thing you could do here is you could use the Biomart library to actually look up each of these ensemble gene names and replace them with the protein name. So it could be a little bit more recognizable as to what each of these were. And then I also looked at the correspondence of TPM and FPKM. And overall, these line up pretty well. If we look at expected count versus FPKM, there's some outliers. And so those might be things that would be interesting to explore more in this data set. Well, why is that expected count different? And you could do that by zooming in on those and then actually naming them on the graph with an ensemble gene ID. So these were just a couple of the examples of ways to use ggplot on the 17 different columns of data in this almost 60,000 genes that are measured in this gene expression RNA-seq experiment. I liked this experiment as a place to start playing with plots and doing data discovery and, really like taking that approach from our textbook of playing with the data as a way to figure out its structure and understand what plots and what tools are possible. This is very similar to the data that you collected in the summer lab. So hopefully it feels really familiar and is something that you are able to tell a story with this week in 3 different plots. Good luck and let me know if you have any questions.